would also like to begin with acknowledging the traditional custodians of this land and pay my respects to the elders, past, present, and emerging. And I'd also like to extend my respects to Aboriginal and Torres Strait Islander peoples. Yeah, so it's really good to be back, and thank you so much for inviting me. Amber said, well, whilst you're here sampling, do you mind talking about? And I was like, no, I don't. I would love to talk about my research. And so I will basically be talking to you about what I do and how the McArthur Basin features, and you'll see why I've called it dynamic. So I'd like you to think about what comes to your mind when you think of the McArthur Basin. You don't have to answer me. I'll tell you what I think, and it's
Gravity. You think gravity?
Yeah, this is what comes to my mind when I think of the McArthur Basin. So this was in 2014 in October, when I was very privileged to have attended an MPGS, and Rowdy led the trip, but MPGS organized the trip and I was part of it. And yeah, I had a really, really good time. I had just started my PhD then, and Rowdy being Rowdy just made this trip really special for me. And I still remember I was going through some of the photos of that field trip for this talk, and I remember presenting on that field trip back at Coates, and there was highlights, there were these field trip highlights, and one of them was beer consumption. So yeah, it was a crazy trip, and I loved it. I learned a lot. Please, I learned a lot, but I also enjoyed a lot. So this is what comes to my mind when I think of the McArthur Basin. So ongoing research. So deep time meaning understanding past ocean chemistry through time, and it's a pretty big task. And I focus particularly in the Proterozoic. And yes, we've got data, but we need a lot of data. So our team at UTAS looks at the concentration of sedimentary pyrite forming in marine black shales. And we've got about 10,000 pyrite analysis, but I suppose we need more, as much as we can, to get a good grasp of what the nutrient concentrations in oceans may have been like in the past and how that fluctuated. So that's a very important part of my research. And I think I should declare it early on that we're quite obsessed with the mineral pyrite at Coates. So we're all doing different things with it. And because I've been indoctrinated as well at Coates, I'm also quite obsessed with the mineral. So I've been recently using that for biogenicity tools. So organisms that have been replaced by the mineral pyrite. Yeah. So you can see the primary textures. Is there any difference in the way that the trace elements are spatially correlated in the fossilized pyrite versus a normal pyrite grain? And if that's true, if you can statistically prove that that sort of, you can use that information, it's geochemical information in my case, can you then use that for applications for other signatures of life in other minerals? So that's something I'm looking into. Stromatolites, because we've got a lot of good data on temperature through time. We've got a lot of data, as I just mentioned, through nutrient availability through time, which is also through pyrite. Can we use the trends and again use stats to see if that has any bearing on available data on stromatolite abundance through time? And there is, and it's pretty exciting research. I'm not including that in my talk because we'll be here for ages. But a crucial concept that came out of my PhD was the stress hypothesis, and I will be talking to you about that in my slides later on, and of course exploration. So yeah, looking at really fine grain, most industry people would think that's boring as, but it's really important for my fundamental deep time research. Looking at pyritized versus unpyritized ammonites, just to begin with, to see if the biogenicity tools can work, and then we can apply it to other things. Yeah, so stromatolites as well, looking at trace element trends through time, combining that with temperature, and then looking at stromatolites. Stress hypothesis, looking at the origins of the first complex cell, evidence of which is plentiful in the McArthur Basin. But this was the most important part of biological evolution, without which there would be nothing on Earth today. So, and the McArthur Basin is a heaven for that sort of research. And of course, exploration. We're all aware, I don't need to tell anyone how important exploration activity is in the McArthur Basin. Thank my team up front. I've learned a lot from these here. People that I've worked with over the years, and that's begin with the photographs aren't arranged by any means in order of intelligence or anything, but they are arranged in how obsessed we are with the mineral pyrite, starting from Professor Ross Large there. I think he started it, and now it's infected us all. And so what he actually did was, he and his team at the time, is to propose sedimentary pyrite trace element concentration in black shells as a new proxy for deep time ocean atmosphere evolution. And this was new, but what isn't new is that people do already look at redox sensitive trace element concentration in black shells, but in home rock. Whereas this was just looking at one mineral and using in situ analytical techniques compared to bulk rock. So it's a very well-established concept about how redox-sensitive concentrations of marine rocks can give you insights into past ocean chemistry. But we proposed, based on what we see in our data, that it's the trace element concentration of sedimentary pyrite that hosts most of the trace elements, and hence analyzing that and looking at that gives us a better idea. And more, because we can then texturally categorize the different textures of pyrite and measure them accordingly. But when you crush the whole rock, you're analyzing everything. So there is a huge difference between that, and it's just another way of looking at deep time ocean atmosphere evolution. And it was also proposed that selenium concentration in particular in a sedimentary pyrite is a good proxy for oxygenation. And so because he started that, and now we're all in this pyrite group that we just love producing lots of data. And so my PhD basically started with you have to work on a lot of pyrite and analysis, etc. Better read up on pyrite. What do you know about pyrite? And I said, well, a lot, because I did my master's. He's like, well, there's a book by David Rickard on pyrite, so better read it. And I was like, yeah, for sure. And then I came across this, and this is the motivation of my research. I'll read it out. The trace element spectra of pyrites and sediments have been the subject of innumerable investigations with a view to using these as environmental indicators. Unfortunately, it is a common PhD topic suggestion, and students are advised to steer well clear of accepting this subject for their PhD studies. The problems are discussed in chapter 13.7.8, which I never went to. I just ran up to Prof's office and I was like, what? Did you read this? And he's like, yeah, well, you've got to figure it out now. And so, yeah, it was quite a, I'm pretty, find it funny now, but I can tell you I really didn't find it funny at the time. So that is the motivation of my research. And so with pyrite, we've been using it for exploration. Really good, brings us a lot of money. And also, like I said, redox evolution and life. And so I'll start with that first, and then I'll end with the exploration side of things. So pyrite chemistry as a biogenicity stool. I will just go through very quickly, but if you like at any, we'll have time for discussion. So looking at a non-pyritized ammonite versus a pyritized ammonite. So what we do is to look at the spatial correlation of trace elements within the fossilized area compared to non-fossilized area. How do we know that? So we do really detailed backscattered electron imaging, and it might not look like much to you, but when you zoom in, you can see that some of the primary, and they're all pyrite, but everything is pyrite, but the textures are still really well preserved. And so the assumption here is that anything, when it's instantaneous biological replacement, there is a sense of ordering within the fossilized part compared to a more random precipitation of the mineral. And so if you can statistically define that in terms of clustering, then you'll be able to develop tools. So yeah, you can see how amazing these textures, primary textures, can still be preserved. So you start off with mapping. Well, you basically go through some SEM analysis first to define your textures. Then you image them and get the trace element data, which is what works with the software. My collaborator was Cooper, he's a statistician. And so we then select regions and look for these clusterings. And so the red box here shows when it's above the 95% confidence interval. So if it's clustering above that, then it's biological, and vice versa if it's below that. So we're still in the early stages, but we've recently been funded to look at a similar sort of thing for the Tasmanian Horodischia, which is again not the fossil, but it's just the shape's been replaced by something else. And so we're trying to look into applying other, there's a really good paper by David Wasey and Martin Brazier, the 2015 paper, where novel approaches towards looking at these fossils are discussed. So we're trying to apply that concept here. But I'd like to emphasize the time, 1.45 billion years. That's Roper in the McArthur Basin. And it's not unknown, the sheer sort of wealth of information that we have on eukaryotic organisms within the Roper group. It's really well established. So it would be really good to get hold of some samples for analysis as well once we've gone through Horodischia. I'm sorry I have not labeled these, and that's because this is part of my current industry project, and I'm not supposed to be sharing any information, but they're unlabeled, so like, what are you going to do with it? So I thought it should be appropriate. But there are some really interesting features that we observe in our Barney Creek Formation pyrites. And they look very biological, but like I said, just because it looks biological means nothing. We have to have a quantitative way of looking at things, and that will have astrobiological implications as well. As something looks like a fossil, but whether or not it is, especially after it's been completely replaced by another mineral, how do you sort of decide whether or not it's fossil or not? So looking at these intriguing textures of pyrite in the Barney Creek Formation, and I think I'm okay to say that these textures are only there in drill holes that are closer to the ore deposit. And this also makes, so a couple more textures, we just don't know what they are. It's just really intriguing and quite unusual. And again, only available in the drill holes that are close to the ore deposit. This is HYC style mineralization. So because I was doing this as an industry project, but I was also collaborating with Eva Stoikin from University of St. Andrews, who was looking at nitrogen isotope signatures in the McArthur Basin. And we recently published a paper on how close to the vent there is exhalation of bioavailable nitrogen that fuels biological activity. And these are some of the new data. I wouldn't call it a pilot study, but this is first of a kind, where we've looked at nitrogen isotope data from near the ore body and then obviously further away. And the signs are that there are hydrothermal exhalations of bioavailable nitrogen that is quite capable of fueling biological, or create these biological niches. Now, whether that's why we're seeing these biological textures preserved is a big correlation, but it's not impossible. So that's one thing also that's in the back of our head. The 1.6, the Barney time is really, really crucial. And I think my collaborators in China were here collecting samples from the McArthur Basin. And luckily, I got to go and collect their McArthur Basin equivalent and the Yang Liao Rift in northern China. Had a great time, thank God, before COVID, because I don't know, I don't think I'll ever be able to go back there. But it was a beautiful field trip and learned a lot again, and collected the same rock equivalent to the Valkyrie, Barney, and Wollongong. So we've got the Chualingo formation, which is the same as the what's the name, Barney Creek formation, and Valkyrie and Chamoline formation. So it would be great to compare the two because tectonically they're related, the Yang Liao Rift, and that's a lot of Allen's and Professor John's work as well on how the North Australian craton was related to the North China craton at some point. And amazingly enough, at 1.6 billion years, you get these weird traces. I had no intention of sampling them. I went there for my black shales, but there's this one outcrop where the shales had been eroded completely and it exposed the carbonates that were underlying. And you would find these textures. It's the cross section of the image before. So there were plenty of these. But again, whether or not they're biological, I don't know. You have to look into it more deeply. But yeah, like I said, there's a quantitative way of sort of ascertaining whether something is biogenic or not would be interesting. And that would change everything. If you can say that you can see traces and then motile traces, because if they are tracks, then that suggests motility. Earliest signs of motility was somewhere around 540, 600. So that changes everything. So there's a lot of potential to basically unravel evolutionary breakthroughs in the McArthur Basin. The other thing is the evolution side of things, and particularly the evolution of the first complex cell, 34 trillion of which you and I are made up of as well. So that key breakthrough had to occur at some point, and it did during the depositional time of the McArthur Basin, so in the middle Proterozoic. When it happened and how it happened and why it happened, this is evolution is like the most dynamic subjects of all time, and I as a geochemist certainly not in a position to solve it. And I appreciate that, but we all play our part, and I'm just here to tell you the small role I would play, or as geologists we would play, in this complex topic like evolution. So this is Earth's timeline. We're all familiar with it. Just going through very quickly. So we've got all sorts of evolutionary breakthroughs occurring. And this is what everyone makes a big deal about. Everyone just loves the macroscopic side of things going, well, that's really when life started, etc., etc. Really, it started way back. And a lot of people think it was by chance, but I disagree with them. And I think there's a stepwise evolutionary trend that we see in record the trend, which will be much clearer if we looked at rocks like the Barney Creek, like the Roper Group in bit more detail than it has been assessed in the past. So milestones in early evolution, the evolution of the first complex cell. So from a very simple bacterial cell to a very complex eukaryotic cell, multicellularity, you and I are multicellular organisms, origin of sex, all of these key biological breakthroughs occurred in the middle Proterozoic. And so when and in what order? So this is a picture of a complex cell, just like it's tiny, but it's got these different cellular components to it. When and in what order they form, don't know. I leave it up to the biologists. Biologists also look into how the first complex cell would have formed. Was it farming? They think that some organisms were ingested by other organisms, not digested, so they could use it when times were bad, etc., etc. Again, a hypothesis. Was it an endosymbiont theory where two organisms came into some sort of mutual symbiotic relationship where they each benefited from one another? Or was it predation? I mean, you can see examples of this today. So that's an amoeba sucking the guts out of the green algae. And you've got predation preserved in our Roper group. Ample evidence of these predatory interactions, ecological interactions in the Proterozoic. So how it happened can be also, it's speculative, whether it's nicey endosymbiotic or whether it was more hostile predation, we don't know. But why it happened, why something would come into a symbiotic relationship or have a predatory interaction is what we can answer. So the thing where geology plays a role is that the tectonic setting, and that will sort of dictate the nutrient flux in the ocean, that will also dictate atmospheric oxygenation. And so the combination of what the geotectonic setting is, what the nutrient flux, so what chemicals or what elements are you making available to the organisms in the oceans? You've got to think of it as a first time for the very first oceans and the very first land that was weathered to sort of drive the nutrient flux into the oceans. Those were the elements that were available to the organisms then. So why certain things happen the way it did is where we feature as geologists. So tectonics, we've got plenty of data on how tectonic activities, and there's really, really refreshing to see Alan and his group and Peter and explaining the myth-busting boring billion, because it's this very weird term that I really dislike. I don't think it's boring billion at all. So good to see everyone jumping on that camp. And there was this paper published this year. They were talking about how the tectonic side of things would have affected Earth's Middle Ages. Oxygen, everyone loves oxygen. Oxygen apparently is the most important element on Earth. And so, yeah, it is definitely important, but is it the most important factor? We don't know. What about oxygen consumption rates of bacteria under nutrient limited? So there might be enough oxygen, but their consumption might be checked depending on what nutrient concentrations there are. And then you've got another very important driver of life, which is temperature. The temperature of the oceans got a huge influence on the organisms as well. So you can look into all of that. But what I do is look into the significance of trace elements, and that's just simply because a lot of life-sustaining reactions will not occur unless we have catalysts. So the temperature and pressure where life sustains, you can't carry the reactions on those temperature pressure rates. You need catalysts. That's where trace elements feature, where you basically expedite the rate of reactions to the level that it's required to sustain life. And you can't have too much of it and you can't have too little of it. So they play a really critical role in basically all sorts of evolutionary processes, because basic cellular functions and the reactions that start and sustain life depend on these elements. So it's really important for us to understand what the availability might have been like of these trace elements in the past. And so what elements are essential for life other than oxygen? Well, you've got macronutrients, you've got micronutrients. And through my pyrites, I'm particularly focusing on the micronutrients. And what I do for that is I collect drill cores. And yeah, collect what? Thousands of drill cores by now. And then cut it, look at it under the microscope. Textural recognition of pyrites is absolutely key before you can target, because there's pyrite in the rock and you can use it for all sorts of things. You just need to know which one is useful for what. And then, of course, you've got the laser ablation, and that does the magic. And so we published this paper, and this is where I will talk about the stress hypothesis, is that everyone in the past mentioned that there is a decline of nutrient trace elements through time, as you can see, highlighted in the red. So this page, so highlighted in the red dip there, and then you see an increase at about 1400. And yes, there's a dip and there's an increase, and it's not just all low and flat as previously proposed. So that was one revelation. And the other is that just because trace elements are depleted, and this is a combination of some of the bioessential elements, doesn't necessarily mean that it's a bad thing. What we proposed in this paper is that because of this nutrient scarcity, organisms were either coerced into coming into a symbiotic relationship. Why else would two organisms who are perfectly capable of living on their own come together? There must have been an environmental pressure, and we think that the nutrient scarcity may have caused that environmental pressure for organisms to either come in a symbiotic relationship because it was scarce, food was scarce, or predatory. Again, there's some form of stress is required for these alternative path for organisms to adopt to alternative pathways, and then in the process evolve. And when conditions better, they are able to diversify and radiate into various other habitats. And that was the stress hypothesis that we proposed. So basically, the low trace element environmental stress, and the high bit is where organisms were able to diversify, and that is validated by the availability of paleontological information that we have for this time period. So yeah, in other words, no pain, no gain is what I actually meant. So this was published, and we further took this research and added more data and started looking at things from a multi-element point of view, not just one element and see what that's doing, just a combination of the two. Because if zinc's limiting, organisms are capable of using cadmium. If cadmium is limiting, they use zinc. So organisms are smart. So you have to look at co-evolution of elements rather than just one. And that again, this was, we used that, and that was published in Geology last year, where we basically made a first-hand link of the data that we've got with actual trace element utilization patterns by organisms, because we have the data for that as well. So which organisms use what element for what purpose, and how does that match with our data is what we discussed in this paper. And yeah, it was quite interesting, and the work led to some really good collaboration with a biologist, Professor Anthony Poole at the University of Auckland, and we're in the planning stages of doing some experiments where we want to, so there'll be a petri dish. This is a very simplistic form. I'm trying to explain biology, and I'm a geologist, so you can imagine what's going on here. Petri dish, there'll be a media, and there'll be prey bacteria, and there'll be a predator, and we want it genetically, he's capable of doing that, modify the predator so it's cobalt dependent. We're going to try out every element. And the only source of cobalt is the prey bacteria. Whether that sort of dependency enables the amoeba to engulf the prey bacteria in real time, we don't know. So we want to test these experiments out with them. Now I'll talk to you about the other side of things, application of pyrite chemistry to exploration. A warning, I'll be showing you graphs with no axis and everything, just so that you're absolutely unaware what I'm doing and I do not get into trouble. But I think I have to provide all the data to NDGS anyway, so you'll find out soon enough. But yes, as beautiful and humble and non-assuming these pyrite framboids can look, they are super powerful in terms of exploration vectors. And I'll just show you how it started with my PhD. When I just looked at three drill holes just to see if because we were looking at black shells anyway, and then we thought, well, if the black shells are mineralized, then we can't use it for our ocean chemistry stuff because it's all buggered because of the hydrothermal activity. So can we then see if there's a difference between a barren shale and a mineralized shale? And if there is, then can we then use it for exploration? This is just the sedimentary pyrite. Previously, all work was being done for hydrothermal pyrite, which is very useful. And so, yeah, we just selected these three drill holes, and we came up with the conclusion that there's a set of elements that, again, this is assuming you're forming sedimentary pyrite in a water column with no hydrothermal activity, as opposed to the same sedimentary pyrite forming in the water column with hydrothermal exhalation. Is it capable to record these events, and if that can be used as a vector? And zinc, thallium, copper, lead, silver, arsenic, they increase closer to the O point. Surprise, surprise, that's not a big deal. But importantly, there are elements such as molybdenum, nickel, cobalt, selenium that decrease. The HYC pyrites virtually have nickel undetected, cobalt undetected. It's incredible. But when you look at further away distal, you get concentration. And that was really the key, because it's not surprising that you see ore elements close to an ore body. So then the ratio became very important. That if one is increasing, good sign, but it's the other element decreasing. And so this coupling can be really powerful. we did propose these two vectors, but we'll have, we'll probably have 10,000 more data now after this project. So this graph is probably going to look a lot different. But what we proposed in 2017 was that you see the black dots, which is your basic pyrite background, and the three drill holes, the MBX, Lala, Yard, and Murphy 4. I know I've got the color coding wrong. I wasn't that much of an exploration geologist at the time, but I know never ever to highlight a barren hole red anymore. But that was what was published, so I can't do anything. But you can see how the McArthur River has its own little signature up there. And it's, again, you've got high ore elements, that's great, but it's also severely depleted in some of the seawater, normal seawater elements that we have. And so then you use that information to devise vector diagrams. And again, you can see the clear distinction between mineralized and the barren hole. And of course, there will be these overlaps, which obviously can be teased apart further if provided we have more data. And so that got us some monies to continue on. And yeah, I've been very lucky to be a part of this project. I've learned a lot and lots of work on the McArthur Basin. So just reassess. So McArthur Basin is just one of the case studies of this big project. Others are doing other things, but I'm in charge of McArthur Basin only. And so yeah, I'm going to show you these graphs. There's some mineralized drill holes and there are some barren drill holes and elements related to mineralization. The red, you can see I've converted into an exploration geologist, not my color coding, right? So the red ones, you see high in ore elements. This is not surprising. Just pointing out that the black data here is barren from MBX, which we think is barren, but the Glide River Basin might serve as a background for us now, which is why I sampled that. But for now, we're using MBX as a barren hole. And so again, all elements related to mineralization are higher than the barren holes, obviously, for obvious reasons. This was based on a very few samples. This is a COVID data as well. So but this was what we found. And then there are elements unrelated to mineralization, like I was telling you about, the elements that show depletion as we go close to the ore body. And it kind of behaved and misbehaved at the same time. And then you've got, amazingly, I just realized that manganese also, something I probably didn't look at when I was doing my PhD more closely, but realized that manganese also went virtually undetected. But manganese in pyrite is just, I can't get my head around it. Sometimes it'll be very promising and then it'll let you down really badly in other data sets. So it's not a very reliable element. But if you look at the trace element adsorption by pyrite and how that's affected by other minerals precipitating alongside pyrite, then I think it might explain a bit better. And so this really good paper by Grant et al., and they're looking at behavior of trace elements in actively forming tag deposit in the mid-Atlantic ridge. So this is like quite modern, but again, it still gives you some idea about how it works. And they propose that the pyrite chemistry is in fact a really good model that represents the hydrothermal system very well. So that's something I'd be keen to do as well, looking at how the modern day pyrites are recording modern day signatures, whether it's all related or not. And so the adsorption of some of these elements within the structure of pyrite is affected with what else is precipitating with it. That's a really cool paper that describes that in a bit more detail. And so like I said, with ratios, I find them very, very useful. And so you clearly see that there is a definite sign, or you can tease out mineralized or close to mineralized as opposed to further away in distal. And you can apply stats and do all sorts of things. I think the data is looking much better now. This, like I said, it was COVID data, but I'm just limited in what I can show you. But what I can tell you is that there's a lot of data, which is what you need for an in situ analysis, where you get so much variation in analysis. Within one sample, you get a lot of variation, but there is a way of tackling it. You don't see that variation in bulk rock method because the method is not sensitive enough to pick those variations. These variations occur in the natural environment. We just have to deal with it using statistics and more data as much as we can. What next? Well, I've applied for a DECRA with Alan Collins at the University of Adelaide. I'm praying, although I'm not religious, but I'm begging people who do pray so I can get my project started. It's very close to my heart. This is an example of what it'll look like. You've got the past, and again, it focuses on the McArthur heavily because we've got so many sequences that we can look at. And Alan's got a fantastic set of samples as well that he said I could work on. So this is what a 1.4 billion year rock looks like, and this is what a modern day sediment core with modern day framboids look like. So essentially, we want to see what shallow water pyrite does, how it's picking up the water column chemistry, because you've got data on modern day trace element concentrations through geotraces. They know what the water column trace element concentrations are. Now, how effectively is that being recorded by a pyrite that I claim to you today is a very good recorder, is going to be a challenge, and this project might solve just that. But again, instead of just looking at pyrite, we'd also like to see what sulfur isotopes, iron and uranium isotopes are doing, because they all have their own shallow water, deep water signatures. And so it's a multi-proxy approach towards nailing water column properties and how then that can be applied or be used to refine our interpretation of all deep type trends. Because right now, none of us are doing shallow water, deep water. We're just saying ocean chemistry. The ocean is a very big place. And so, yeah, as long as we acknowledge it and then do these groundwork, that'll be great. So please, God, let this be funded. And with this, I come to an end with my presentation. But because I'm in the Northern Territory, you've got to be in the spirit of the Territorians. So I'd like to share some crazy experiences that I had. I was here last time. And yeah, I don't know why I did it. I don't know what I was thinking, but at the time it sounded right. And there was this crop called the chopper. And yeah, this was me, as you can see. I was and they decided to feed a fish whilst I was in there. And I was like, well, I'm going to show this to them and show them how brave I was 6 years ago. And I'm not excited about crocs anymore. You know, been there, done that. And what happens? History repeats itself in 2021 when I learned that Shannon at the core shed owns a pet crocodile. And so I think the roles were a bit reversed. I was in a more powerful position. And you know, I was quite happy, as you can see, but this is how I was really feeling. I was like, come on, hold it for a photo. And Daryl was loving me being just crazy scared, but I was like, yes, all right, all for a photo, I'll smile. But yeah, I loved my experience yet again at the Northern Territory. Thank you.